I want to take a moment and thank you all for coming and bring something to your attention. Some of you are aware, and others may not be, that this is National Graduate Student Appreciation Week. So I just wanted to take a moment and point out something that I know everybody is aware of, is that grad students are an essential component of our program. And I wanted to take a minute to say I am very thankful for everything you do for the program. Above and beyond the formal research and responsibilities, you've all been wonderful about engaging with the broader community, helping out on all of these committees with recruitment. I think we had a fantastic recruitment season. I know a lot of you worked really hard on that, working on the diversity committee, the reading groups, retreat, all the many things you do for each other and for the program in a general sense. I just want to take a minute and say thank you. I'm sure everybody will agree with me.
Thanks, Nicole. So last night when I was thinking about what I was going to say to introduce Josh, I realized, I want to find some embarrassing story or some embarrassing pictures or something like that. And the problem is, of course, that requires memory and it requires organization. And one of which I suffer aging-dependent decline, and the other one is constitutively never been there. So I don't really have any stories. But Josh was a graduate student at Columbia with Gary Stroll, did important work on RNA binding proteins, Bicoid. During development, he moved to Cold Spring Harbor to do postdoc with Tim Tully, and got promoted at Cold Spring Harbor around 2002, right, or three, something around there. And he stayed there for a stretch of time before moving to Stony Brook in 2016 and 2017. So, it's sort of inevitable, I think, that as time goes on, you start worrying less about function and more about dysfunction. And so he's going to talk today about retrotransposons and their role in neurodegeneration.
Thank you, Jerry. Thank you for not sharing any embarrassing stories. I'm tempted to share some embarrassing stories, but I'm going to use my prefrontal cortex and not do it. So what I wanted to tell you about today is some of the work in my lab that has to do with something that we call retrotransposons that we are excited to think has something to do with neurodegeneration. And I thought I would start with three of my heroes on the first slide. So raise your hand if you know who that is. Yay, I'm always happy when people know who Barbara McClintock is. So she is a clear genetic hero. She was one of the most brilliant geneticists in the history of the field, and she discovered transposable elements while studying maize. And this is Lou Gehrig, who is a childhood hero of mine. I'm not that old, but I mean, I've heard stories about him. As a Yankee fan growing up near Yankee Stadium, I always knew about him. And the work that I'll tell you about has something to do with ALS, which in the United States, some people still call Lou Gehrig's disease. And this is another hero of genetics, that's the fruit fly Drosophila melanogaster. So why is this not advancing? There we go. Okay, so I want to start by talking about something that is really one of the most fascinating and I still think poorly understood aspects of neurodegeneration. And that is that most people who study neurodegeneration, most clinicians who see patients would agree that there is what appears to be a focal onset and spread through neural tissue. And across different disorders, Alzheimer's, Parkinson's, ALS, frontotemporal dementia, there are different proteins that are seen by pathologists when they look post-mortem to have undergone misfolding, aggregation, and these can be intracellular or extracellular. But what is commonly thought is that the onset is focal within a part of the brain and progressively spreads through circuits over the course of disease. And that really is something that needs explanation. In the case of ALS, it's probably the most obvious to both the clinician and the patient, because a patient will typically come to the clinician and say, you know, I'm having trouble doing certain tasks with my left hand. And then that phenotype, the dying of the motor neurons and the wasting of the muscles will spread, you know, along the length of the limb that is first affected, and it will spread to the ipsilateral limb, and it will spread contralaterally. It spreads as per the topographic organization of the motor system. And it can initiate in upper motor neurons or lower motor neurons, but ultimately will engage both. And when pathologists look post-mortem in brain tissue from affected individuals, they see a pathology that is observed in both neurons and glia that involves a protein called TDP-43. This is an RNA and DNA binding protein. It stands for TAR DNA binding protein 43. The T, TAR, is the HIV TAR element. It was first identified as a 43 kilodalton protein that binds to the HIV TAR element. And since then, it's been found to bind to both DNA and RNA. Its normal localization is in the nucleus, which is shown in blue. And in pathological tissue, a pathologist will find cells where it has been cleared from the nucleus and accumulates in these large cytoplasmic inclusions. And they typically observe this using antibodies that detect a hyperphosphorylated form of the protein that accumulates within these pathological inclusions. And as I said, that's seen in both neurons and glial cells. Now, all of these disorders are genetically and clinically complex. And just to give you a flavor for that, there are single gene mutations in many individual genes that can cause ALS or frontotemporal dementia. And both FTD and ALS lie on a symptomological spectrum. So there are cases that have of frontotemporal dementia with motor neuron involvement, and there are patients with ALS who have dementia symptoms. And there are mutations like C9orf72, an expansion repeat, that can cause both ALS and frontotemporal dementia-like symptoms. And there are mutations like SOD1 that really only cause ALS. But only about 10% of the cases of ALS, for example, are thought to be familial. The overwhelming majority, over 90%, are thought to be what's called sporadic. And when you look in ALS or frontotemporal dementia, when pathologists look, they see a heterogeneity across the disease in terms of the protein pathology. In ALS, although only a tiny fraction, so a tiny fraction of the 10% that are familial have mutations in TDP-43, and yet 97% of the patients have that pathology that I showed you on the previous slide. In the case of frontotemporal dementia, it's about 45%. Now, let's think for a minute about this focality and spread. On this model on this slide is the model that is probably the preferred model in the field. It's what many people assume to be the explanation for spread from cell to cell. And this idea really is that these aggregation-prone proteins, SOD1, TDP-43, many others, have low complexity domain regions. They are prone to aggregation, and they are thought to undergo prion-like conformational changes, whereby a misfolding then seeds recruitment in trans of other copies of that protein to misfold. And then the idea that's proposed is that this is not only toxic within a cell, but can lead to release of seeds that are then taken up by a neighboring cell, reinitiating the cycle of pathology. And that this is the model that has really been proposed by many people in the field to explain the spread. This idea comes from Prusner's work with transmissible disorders such as the mad cow disease that you will all have heard about, and equivalent disorders that affect many species, including people. These are infectious prions, infectious proteins. They have the property that I described on the last slide. And the proof that they really have that property is that one can take misfolded PRP, the protein that is encoded by the PRP gene, and inject it into a mouse. So this can be purified from the brain of a sick animal, inject it into a mouse. Over time, which can be months, the seeding occurs within the brain of the injected animal. The animal is dead, and seeds can be then purified and can be passaged and re-injected into a new animal. And importantly, there's a species barrier, because if one takes misfolded protein from hamsters and injects it into a mouse, there is no cross-seeding, or very, very little, very rare, and the animal survives. But if one injects the hamster protein into a mouse that is transgenically carrying a copy of the normal hamster protein, you again recapitulate this seeding property, prion misfolding, eventually death, and one can then passage from that animal. That's really different from ALS and Alzheimer's and frontotemporal dementia and Huntington, which do not have that property. There are remarkable findings in the literature, such as the one described from this very nice paper from Trojanowski and Lee labs. And what they did is to purify seeds of TDP-43, misfolded prion-like seeds of aggregated protein from a frontotemporal dementia patient, propagate that in mammalian cell culture, in a cell culture that expresses the human gene, and then inject that into a mouse, into cortex. And this is a transgenic mouse that pan-neuronally expresses a copy of the human gene that is mutated so that it is already mislocalized to the cytoplasm where it's prone to aggregation. And what they observe is, if you zoom in, using antibodies like the ones that pathologists use to look for hyperphosphorylated protein, they see that it is taken up by cells and forms these cytoplasmic inclusions, abnormal distribution of the protein and hyperphosphorylate. And when they look in contralateral cortex, presumably because of projections through the corpus callosum to the other side of the brain, they observe fewer, but they observe cells that have taken up some of this protein and appear to have propagated the pathology, because remember, this is a transgenic animal that has the human protein. But importantly, so they can inject the seeds, they visualize with this antibody, they see the pathology spread. But importantly, these mice express a pan-neuronal transgenic human form of the protein, and they do not observe spread of the cellular degeneration. It's not like PRP. These mice don't die, and you can't then pull extract from these and inject them again into the next mouse and propagate it. So it's a little bit different. There's a commonality here, but it's not exactly the same as an infectious disease. And I want to take a minute before I introduce the data I'm going to show you and just formally think about the fact that there are two categories of model here. The first category of model is Prusner's model, which is that some upstream signaling event, it could be a mutation in the gene, or it could be some spontaneous thing, or it could be eating, an infected brain, if in the case of the actual transmissible prion disorders. Some upstream event causes aggregation. In this case, with TDP-43, it would be in the cytoplasm. And the idea is that seeds of this are released and taken up by another cell that then templates in this prion-like model a misfolding, leading again to release and taken up by the next cell. That's the prion model. But there's another model, which I'll call sequential feedback mediated intercellular signaling, which in the case of TDP-43 and the experiments that have been done cannot be distinguished from this model, but is mechanistically very different. So far, cannot be distinguished. And in this model, you have some event leading to aggregation, leading to release of some factor X. So this aggregation may stimulate factor X and cause it to be released. And factor X is then taken up by a cell, and X then causes some signaling events in that cell or some other events that lead to aggregation of the protein TDP43 in this case, and that's propagated. And in this model, aggregation is upstream of X, and X is upstream of aggregation, and therefore it's a feedback. And in my opinion, this is a more robust mechanism for propagating a pathology through a tissue. So I just want to start with just formally thinking about those ideas and then present the data that I'm going to show you. Oh, and one more thing. This propagation or this pathology, let's call this the non-cell autonomous effects that are observed in this disease are not only neuron to neuron, there's also tremendous involvement of glial cell types. And so this is now a classic experiment of primary astrocytes from ALS patients grown in co-culture with motor neurons, or even with conditioned medium from the primary astrocytes from ALS patients. This experiment from this paper is conditioned medium. And what they show is if they take primary astrocyte conditioned medium from healthy controls or from ALS patients and put that onto motor neurons, that the conditioned medium from the astrocytes from ALS patients impairs the survival of the motor neurons. So I want to propose a model, which I'm then going to show you some of the evidence for. And the idea is that TDP43 protein aggregation synergizes with the effects of age to cause a morbid loss of the silencing systems that normally stifle the expression of retrotransposons and endogenous retroviruses. And I'll unpack what that means, resulting in a storm of their expression and or replication with myriad toxic cellular effects leading to death. But I also want to propose that this effect has a local onset followed by spread through the CNS in a non-cell autonomous and self-amplifying toxicity. And I want to tell you today what I think the mechanism of that is. So what are transposons and what are retrotransposons? There are two types of transposons. The first we call cut and paste, or DNA elements. And the second ones we call copy and paste, or so-called retrotransposons. For the fly people out there, these are like P elements. And for the plant people out there, these are the Barbara McClintock ones, the ones that she first studied. These are DNA elements that cut themselves out of chromosomal DNA by a recombination-based mechanism and paste themselves into a new location. And that is not the type we're talking about today. They're not thought to exist in active form in humans, although they do in flies. We're talking about retrotransposons. These exist in the DNA like a provirus, like HIV, like a retrovirus would. The provirus codes for an RNA that goes into the cytoplasm where it encodes for one or more proteins that bind to the RNA and catalyze a reverse transcription reaction, cleave chromosomal DNA, and reinsert at a de novo location. These are evolutionarily related to retroviruses. In fact, the LTR retrotransposons are the evolutionary ancestors of retroviruses. All retroviruses evolved from these elements, and retroviruses have also reinfected organisms and become endogenized again to become retrotransposons, and they're often called endogenous retroviruses. And so there are autonomous and non-autonomous elements. These are fully functional. These require the proteins from those. The autonomous retrotransposons consist of line elements and LTR retrotransposons and endogenous retroviruses. There are millions of these in the genome. There's a few hundred, maybe 100 or so of these in the human genome. And although there are many of these types, HERV K is the most closest to fully functional in humans. It can be expressed, various copies can make each of the open reading frames. It makes virus-like particles and so on. And these together make up almost half of the human DNA content, most copies of which are not fully functional, but as I said, some copies of which are. And when they are expressed, they can be highly toxic. They can cause DNA damage leading to apoptosis or cellular senescence. They can cause insertional mutations. The RNAs and cDNAs and proteins that they make can be toxic to the cell. And in particular, the RNAs and cDNAs may activate inflammatory signaling. So I want to take you through some published literature so I can tell you an unpublished story. I'm just going to summarize here. There is now a pretty robust literature from my own lab and from a number of other labs establishing that normal aging leads to an erosion of the systems that normally silence retrotransposons. We showed this in Drosophila brains, but many other papers from Steve Helfand and John Sidivi and many others have found this to be true across species and tissues. Knockout or overexpression-driven pathology of TDP-43 leads to a collapse of post-transcriptional silencing systems for retrotransposons and probably chromatin level ones as well, leading to broad overexpression and de novo replication in neurons and glial cells. The endogenous retrovirus MDG4, previously referred to by the name gypsy, which I will try to avoid saying because it is a slur against Romani people, I will try to switch to this name, mediates much of the cellular toxicity of TDP-43 in Drosophila. I'll show you one or two pieces of data from those published studies. And now there's a literature showing that in human tissue from patients, retrotransposons and endogenous retroviruses are broadly overexpressed. That's true from ALS, frontotemporal dementia, as well as from tau pathologies, including Alzheimer's and PSP. And as I mentioned, these are low complexity domain proteins. There's a whole literature on how this may lead to aggregation pathology. The main thing is that the aggregation pathology that is seen in patients includes loss of nuclear localization. And although the accumulation of inclusions in the cytoplasm may contribute to toxicity, it is now widely thought that the loss of nuclear function is really important here. And we have found, and others, that both overexpression driving pathology and loss of nuclear localization or knockout of the gene are each sufficient to lead to activation of retrotransposons. And this is what this loss of nuclear localization looks like. When we drive it in Drosophila, the green signal is an antibody. This is actually human TDP-43 with an antibody that pathologists commonly use on postmortem tissue, and we see it accumulating in the cytoplasm and excluded from the nucleus. And a number of years ago, we found that driving TDP-43 leads, this is RNA sequencing, leads to pretty strong upregulation of many different retrotransposons across all of the types of retrotransposons that exist in the genome. And in collaboration with Molly Hamill's lab and a number of other collaborators, we found that in a subset of ALS patients, if you look in cortical tissue, motor cortex, there's a subset of the patients that have what I would call the fly signature. And these are the patients that have the highest degree of TDP-43 pathology. The fly signature meaning overexpression of endogenous retrotransposons. In fly, we are able to do manipulations to demonstrate some causal impact. This is lifespan, percent survival over age. Median survival of 63 days in this experiment is the wild type. When we drive TDP-43 in either neurons or glia, in this case glia, we dramatically shorten the lifespan. And if we just knock down the MDG4 retrotransposon using an RNAi transgene just in those glial cells, we have a dramatic, although partial, restoration of survival. And we think this has to do with DNA damage, which is part of the replication cycle, because when we knock out CHEK2, which is a sensor of DNA damage that signals for apoptosis, we are able to almost completely restore survival. So these animals here, this population, have overexpressed TDP-43 in glia, but have the CHEK2 signaling protein knocked down with RNAi. And so we constructed a replication reporter that I won't go through because it's published. For the virologists in the literature, you'll be able to look at this cartoon and figure out how it works. It turns on mCharity in the nucleus, and this version turns on GFP at the cell membrane only after the retrotransposon, or I'll call it a virus because that's really what it is, replicates through an RNA intermediate and reinserts into chromosomal DNA at a de novo location. And when we make a transgenic animal of this, what we observe is that in the animals that we induce TDP-43 at time 0, this shortens their lifespan, and we see a dramatic increase in the M-cherry signal, which denotes replication of the reporter. Is there a way to turn the lights down? Let me see if I can do this. Oh, wrong ones. I want to turn the ones down up here. That's probably better. It's too dark. Hang on. You guys are going to fall asleep. That's good. Can you see that red signal here now? Yeah. So that's the M-cherry signal. This is induction, just in glia, and looking 2, 3, 4, or 5 days after induction, and you see the accumulation of this red M-cherry signal that denotes replication. This is happening in the glial cells because that's where we're expressing TDP-43, but the reporter is also only being queried in the glia for technical reason. And the white marker here labels the glia, and you can actually, the glial nuclei, and you can actually see that the glia are lost over time. And when, this is done by a postdoc in the lab, Yonghan Chang, who we call Forever, that's his English name, who by the way is on the job market, and he's fantastic. When he looked in close using a marker of DNA damage, he can see that the individual nuclei that exhibit DNA damage are the very same ones where the retrotransposon reporter has replicated. Cell by cell, almost without fail, the ones that have the replication of the endogenous retrovirus have DNA damage. They also exhibit activation of caspase-3 signaling, indicating that they're committed to cell death. And in fact, what Forever was able to do was use a recombination-based system called the FLIP system to turn TDP-43 on in little clusters of cells, in this case, a cluster of astrocytes. And this white label in these neurons nearby, the neurons are labeled in blue, the nuclei of the neurons, this white label says that they've activated caspase-3. So these neurons are dying because they're near these astrocytes that have TDP-43. This is a surface glia, part of the blood-brain barrier, called an SPG. These neurons are dying because they are near this glial cell that expresses TDP-43. And if he merely knocks down the expression of the endogenous retrovirus or of the CHEK2 DNA damage sensor within the cluster of cells that overexpress TDP-43, they no longer kill the nearby cells. And so this led to a model, which I now want to expand upon, which basically said that there's a cycle in which TDP-43 inclusions form disrupts the siRNA surveillance system, prior publications that I didn't show you, leading to activation of these endogenous retroviruses, which may cause DNA damage, and that somehow this cycle leads to release of something toxic that kills nearby neurons. We knew that it caused more DNA damage there, but we didn't know what the mechanism was. And we didn't know whether these toxic factors were actually movement of the seeds of TDP-43 misfolded protein, release of some inflammatory cytokine, or movement of the retrotransposons due to their viral-like nature. And what I'm going to tell you in the rest of the talk is that this is playing a major role. So what Forever did in order to test this was to create an interesting context. He took a transgenic fly that has human TDP-43 with a flag tag, wild type TDP-43, knocked in to replace the endogenous fly gene. Now, when you knock the fly gene out, it's lethal. This is fully rescued by the human gene. They have no evidence of neurodegeneration, no evidence of TDP-43 pathology. They look totally normal. And I'll show you some of that. And then on this background, he added various fly tricks to induce cell type specific overexpression of human TDP-43 just sparsely in one subtype of glial cells to induce pathology here and ask what happens nearby. So when he does this, we get the aggregation pathology here that I'll show you, and we can assay the nearby cells. And just to show you the effect on lifespans, This is the wild type lifespan. One of these two curves have the human gene knocked in to replace the fly gene, and the other one has that plus the various GAL4 and GAL80 genes to induce within glia, but no UAS TDP43 gene. And then this one here has added induction just within a subtype of glial cells, and that is sufficient to actually affect the lifespan of the animals. And it also leads to a spreading pathology that I want to describe to you. So he's taken a confocal stack from a part of the brain that's cartooned here. In blue, the neurons are labeled with a marker that labels the neuronal nuclei, that's ELAV. The red ones, which are not all labeled here, are the glia, and in particular, he's going to turn on TDP43, human TDP43 at higher levels to induce pathology in this type of glial cell called SPGs, which are at the surface. That's here. The green label is hyperphosphorylated TDP43. That's pathological, that we first see appearing in the nucleus, but we also start to see puncta in the cytoplasm. And over time, more and more of it accumulates in the cytoplasm. At a distance from that, in these neurons, we begin to see hyperphosphorylated TDP43. And that does not occur in the animals that just have the basal level of human TDP43 in all cells without the induction in this surface glial cell. And when he zooms in on the neurons that have phosphorylated TDP43 in the nucleus, they invariably begin to lose the flag tag, which marks the humanized locus that is knocked in and expressed at physiological levels in all cells. And this is at 7 days induction. It becomes even more apparent at 15 days. You can see here's the phospho-TDP43 within the glial cell. You see a lot more moving out into the cytoplasm. Many more neuronal nuclei are now beginning to have phospho-TDP43 and are beginning to have a diminution in the flag tag. He came up with some metrics to quantify the distance of spread. So what we decided to do is just treat each neuronal nucleus row as like a row number 1, 2, 3. And he measured the distance from which each cell was counted for what distance in diameters of cells it was from the surface glial cell. That's shown here. These are all controls that have, for example, the human TDP43 knocked in, but not pathological, no phospho-TDP43. By day 7 or 15 after induction in the surface glia, you begin to see within this optical section increasing distance from the original initiator cell, phospho-TDP43. The other measure is the percentage of total neurons in the field that are labeled. And again, you see day 7 and day 15 increasing total percentage of neurons that are labeled with this phospho-TDP43. And these neurons are dying. This is quantifying the total number of neurons. And you can see that relative to controls, by day 7 and 15, you actually have a loss of the total number of neurons. We also see propagation of DNA damage. So this yellow marker is the gamma H2AV. He's inducing in surface glia here TDP43 pathology, and we actually see the DNA damage spreading as well. And that's quantified here. So the same layout as before. This is number of rows spread, and this is total percentage of neurons that have DNA damage, only increasing in the experimental groups and higher 15 days than 7 days after induction. Importantly, Forever also tested what happens when there is not a knock-in. So this is a case now where the fly TDP43 protein is expressed. The antibody doesn't detect that protein, but he induces human TDP43 in the surface glia, and this is, he co-induces GFP, so you can mark the surface glia, and TDP43 fused to RFP, so you can visualize it independently of the hyperphosphorylation marker. And here he sees, the blue is the hyperphosphorylation, the nucleus and in the cytoplasmic puncta, he sees hyperphosphorylation, pathological TDP43 within the surface glia. but no evidence of spread. Now, the antibody doesn't detect the fly protein. We can't say whether the fly protein is aggregating, merely that we cannot see the spread of the phosphorylated human TDP-43. And that's a zoom in to show that, to show, I think it's this region. And when he looks at the quantification of this, there's no evidence of spread. But when he looks at neurons and he counts the number of neurons, there is a loss of neurons that is just as bad as when we do this in an animal that has the human gene knocked in. So either something else is spreading, or there's no species barrier to the cross-seeding. And I find that hard to believe. In other words, if aggregated TDP-43 movement from cell to cell is underlying the non-cell autonomous killing of neurons, then you would have to postulate that the human misfolded TDP-43 moves to neurons and is perfectly effective at cross-seeding the fly protein to aggregate. And I have a hard time believing that that's true, and I'll show you why we think it's not. So that I just said that, and that's what I said. Now I'm going to show you that this spread requires expression of the endogenous retrovirus in the surface glia. So now Forever is labeling all the neuronal nuclei in blue. All the glial nuclei are labeled in yellow, and phospho-TDP-43 in green. So here he's expressing, again, high levels of phospho-TDP-43 in the surface glia and observing spread of that pathology to neurons. This has the knocked-in humanized allele in the fly locus. And that spreads both to neurons and actually to some of the other glia that are not on the surface. But when he knocks down MDG4, this endogenous retrovirus, and knocks it down only within the surface glia, he greatly reduces the spread of the phospho-TDP-43 appearance in the neurons. And that's quantified here. So this is row number of spread at day 7 and day 15, and now day 7 and 15 with MDG4 knocked down. So the average number of rows of spread is decreased. The result is more dramatic when you look at the percentage of neurons labeled with phospho-TDP-43. This is day 7 and day 15. This pointer is dying, but I hope you can see the next bars are reduced because of knockdown of MDG4. Total number of neurons are also rescued. So this is total number of neurons at day 7 and 15, and increased when you knock down MDG4 in the glial cell. So from these experiments alone, we cannot determine whether the appearance of TDP-43 proteinopathy in neurons involves movement of pathological TDP-43 seeds from the SPG glia to the neurons. What we know is that it requires that the human flag tag TDP-43 is expressed in the neurons. We know that it requires MDG4 endogenous retrovirus expression in the SPG glia. And importantly, the toxicity of human TDP-43 overexpressing glia to neurons that expresses physiological levels of TDP-43 is equally severe regardless of whether the neurons express human or fly TDP-43. So what Forever did next is to utilize a reporter system that a former student in the lab, Rich Keegan, produced, and that is, so this is basically a retrovirus. It has LTRs, and it has open and reading frames for GAG, POL, and ENV. And what Rich and Forever did is to tag ENV with mCherry using a P2A peptide between, or to just delete ENV and tag with mCherry. And then what Rich Keegan showed is that if you transfect that into one population of Drosophila S2 cells and mix those or put them in a transwell system, which I'm showing you here, so that the transfected cells are on the top and the untransfected cells on the bottom, or the other way around. And then you image, after co-incubation, This is a transwell with a membrane that has a 0.4 micron pore size, so cells cannot move, but viruses could. And if you incubate them for several days and then image the cells that were untransfected on the bottom, he can observe movement of the M-cherry signal that was attached to the virus. But if he deletes env, it can't move. And if he co-expresses env in trans, it does. So that's published, and I'm not going to take you through all the data. That's some of the data. So it's an endogenous retrovirus. And I want to point out that transfer between cells may actually be a general phenomenon of retrotransposons and endogenous retroviruses. Even with line elements, the top line, there's some evidence for horizontal transfer in extracellular vesicles. But with both the endogenous retrovirus MDG4 that I just showed you, and with HERV-K, there's evidence for formation of virus-like particles and movement between cells. And so we wondered whether the toxic factor that moves could actually be a viral transfer of this endogenous retrovirus. So Forever did some further experiments in S2 cells. On the left, he's co-transfected S2 cells with a nuclear M-cherry and with human TDP43 that has a V5 tag, so we can independently look at TDP43 with the V5. They both go to the nucleus as shown in most of these cells, like here and here. Occasionally, he sees a cell where the nucleus is labeled, but there's also this what we think is an inclusion of TDP43 appearing in the cytoplasm. That's the effect that's well known from overexpressing this protein. It takes time, and between 24 and 72 hours, he begins to see more and more cells like this. But if he co-transfects instead of M-cherry, the MDG4 retrovirus tagged with M-cherry, he sees that the majority of these cells start to have these puncta in the cytoplasm. That's quantified here. So at 24 hours, about 40% of cells have some evidence of TDP43 appearing in the cytoplasm, but over 60% of cells if they're co-transfected with MDG4. So the retrovirus is accelerating the cytoplasmic accumulation, and he's also shown that it's hyperphosphorylated. And now what he did is to transfect separate populations of cells. On the left, he transfected one population of cells with M-cherry and one with human TDP43, grew them long enough for them to express, washed them, and mixed them. And you see that the M-cherry stays where it should and the TDP43 stays where it should, and they don't mix, they don't move. But when the M-cherry is attached to the MDG4 retrovirus, he sees M-cherry in the cells that are transfected, and occasionally he sees nearby cells that have taken up the M-cherry signal, and that is a cell that expresses TDP43, and invariably it starts to accumulate in puncta in the cytoplasm. If it has taken up the M-cherry signal, TDP43 accumulates in cytoplasm, and that's quantified here. So at both 20 hours after transfection and 24, there's this increase. So first of all, between 20 and 24 hours, there's an increase in the movement of TDP43 in the cytoplasm, and by 72 hours, it would be even more. But this is accelerated in the presence of MDG4. If the cell has taken up the MDG4 retrovirus, then it increases the chance that TDP43 is in the cytoplasm. And finally, he did this transwell thing, and here he's imaging the cells on the bottom that were transfected with TDP43. The cells on the top are transfected with either M-cherry or M-cherry attached to the virus. So here, I love this one because these are two cells next to each other from the bottom. This cell took up the virus and this cell didn't. Look at the difference in distribution of TDP43, and the yellow now is hyperphosphorylated TDP43, showing how clearly it moves out into the cytoplasm and that it's taking on this pathological form. And here's a few more examples of cells that took up the virus compared to controls on top. And that's quantified here. So we have this basic idea, and I'm not saying that this is wrong. I'm not saying there's clear evidence that these prion-like proteins can move between cells. That's observed. The question is whether that is sufficient to explain the spread of pathology. And I've always been struck by the fact that you can't serially passage like you can with PRP, that the animals that you inject these things into don't die, and that even the cells that take it up have not been shown to have much pathological impact. And what I've shown you today is that there is, so in previous work, we showed that inducing TDP43 proteinopathy or knocking the gene out is sufficient to cause activation of expression and even mobilization of retrotransposons and endogenous retroviruses. But what I've shown you today is not only does TDP43 pathology stimulate these mobile elements, they stimulate TDP43 pathology, and they can move between cells. And what I would say is that given this feedback amplification, movement of either a seed of TDP43 or the endogenous retrovirus or a retrotransposon would be sufficient to reinitiate a feedback amplification loop that I'm proposing could explain spread of pathology through neural tissue. And I want to end by thanking the people who did the work. All of the unpublished work I told you about today was from this incredibly talented postdoc in my lab, Forever, who's on the job market. The work in cell culture to generate the reporter was done by Rich Keegan, although the work that I showed you at the end that's unpublished using that was done by Forever. These are some former members of the lab who were involved in earlier stages of this work and current and recently departed people. And finally, let me acknowledge funding sources. I'm happy to take questions. Yeah.
Oh, sorry. So because you could do this thing with S2 cells, there's really no cell specificity, cell type specificity. Is that correct? Why did you express TDP43 glia and visualize transfer to neurons?
Yeah. So I don't think there's, I don't think I would agree that there's no cell type specificity. What's interesting is so there's two questions. One is there cell type specificity to the virus, to the endogenous retrovirus? Nobody knows what its cellular receptor is. And it may be that S2 cells, macrophage lineage, just happen to have that and we got lucky. We don't know in tissue. We've never shown yet that the retrovirus moves between cells in tissue. It's much harder to do for technical reasons, but we have various flip design construct to ask that question. The question of cell type specificity in vivo, the reason we looked in glia is, so when we first did RNA sequencing in a 2017 paper on Krug et al., where we drive TDP43 either glia or neurons, and we take the whole head and we do RNA sequencing, we found much more robust effects on retrotransposons in glia than neurons. Although there were ones in neurons as well, they tended to be different ones. MTG4 was in glia. And so it looks like it is induced mostly in glia. And we had reagents for that one. It seems to be a major player. But there may be other elements that are induced in neurons, and we just haven't touched that yet. Yeah. Or even cell type specificity within neurons.
Great talk, Josh. A couple questions. One's technical. So when you use these TDP43 clones that are expressing in the glia, and you see the expression in the neighboring neurons, how do you know it's not just leaking a UAS cassette expression in those neurons versus transfer of protein?
It's a good question. So we can do the same experiments where we're just turning on fluorophores, but not TDP43, and we don't see the fluorophores move. We also have used this approach. I didn't say this because it's in the paper, but we've used that same approach instead of turning on TDP43 to turn on pro-apoptotic genes, and we can successfully kill the glial cells, but it doesn't kill the neighboring neurons. And then finally, we can express TDP43 in glia and overexpress inhibitors of caspase, like DIP or P38. And they block caspase function that actually prevent the glial cells from dying. The glial cells now stick around, and it's way worse for the neurons. They just
die like crazy.
Yeah.
So we really think they're releasing something. Yeah.
And the second is sort of a follow-up to Jerry's question. So the spreading idea is a really cool idea. I'm still not fully convinced that the field's kind of demonstrated that.
You mean the spread?
Well, I think there's other explanations for the spreading, for the apparent spreading, of ALS that don't involve the protein seeds and protein transfer.
Yeah.
But the question is, why specificity for motor neurons?
Those are great. Sorry, was there another?
No, that's the,
Yeah,
there's still this fundamental question of why is it that
Yeah.
motor neuron is affected?
yeah, those are all great questions. So I'm not convinced about, so I think that there's reasonable evidence for the spreading of TDP-43, for the appearance of TDP-43 proteinopathy in different cells, with one exception. I'll say one thing about that. In that experiment that I showed you, which is probably the best of the papers, they inject into cortex and they look in contralateral cortex, but projections run both ways. And so what I think is hard to rule out is that the injected protein is taken up by axons and then transported back. So in that case, it might not actually be spread. So I kind of agree there. But if we give them that, they haven't shown that that's really what causes the spread of disease. It's the movement of the protein. I also think that, and you might have been alluding to this, it's worth acknowledging that there are other models out there. The other model that I actually find to be somewhat attractive is that it starts with death of inhibitory neurons in motor cortex, and that leads to hyperexcitability of upper motor neurons, which may drive glutamate toxicity in motor neurons. The nervous system is connected. There's lots of ways for non-cell autonomy. And I don't rule that out, and I think that's a really interesting idea. And the final question was specificity for motor neurons. I mean, if you look at TDP-43 pathology, I mean, I'm told by pathologists, because I don't do this, you actually at terminal stage often see it throughout cortex. So I think the specificity is a bit overstated, but maybe there's some heightened sensitivity to it in motor neurons, and it's not as specific as it might appear.
So
that kind of answer the
in that
question?
case, they're more sensitive to the effects of aggregation as opposed to being.
That's possible. That's possible. I mean, the problem is that, in patients, we get end stage. In animals, we can look along the way, but animal models are not people. So we're kind of touching parts of the elephant and then trying to draw the whole picture. Yeah. Sorry. Yeah.
Seems like a really unfortunate coincidence, this feed forward with that TDP-43 dysfunction allows retrovirus activation, and retrovirus activation leads to TDP-43 dysfunction. And I'm wondering how you're thinking about that, and if perhaps there's a different cellular context, maybe during development, where this is an advantage for cells, that it's like activating the immune system to kill cells where retrotransposons have been activated.
Yeah, that's a really interesting question. I mean, the way I think of it is, I mean, TDP-43 didn't evolve to cause neurodegeneration, right? I think of it as part of the cellular surveillance against viruses and endogenous retroviruses and retrotransposons. My feeling is that outside the brain, the best thing for a cell to, in a replenishing tissue, the best thing for a cell to do when these things don't become active is to die. And maybe to release, and maybe to attract the immune system and say, kill me, right? And start a whole inflammatory response because you want to get rid of infected cells or cells that have an endogenous, activation. In the brain, that may be problematic, but evolution might not care so much what happens to us at our ripe old ages. So that's kind of my hand wavy. You buy it? Yeah, okay.
Kind of following up on what Claire asked, I was wondering, is there some other way to kind of decouple that feedback loop? Is there some other mechanism that activates the retrovirus and then look
Yeah.
at TDP without having that been the stimulus.
Yeah, no, it's a great question. That's aim 3 of my grant that probably won't get funded. Is to basically to directly drive retrotransposons, transgenes to drive them. But to do that, with cell type specificity, so we can then observe what happens in neighboring cells. And the other is cell type specific knockout of genes that normally control retrotransposons. Reviewer 3 thought that those were not very specific manipulations, knocking out, organoid proteins and things like, and they're right, it's not. I mean, it could affect other aspects of RNA biology, but I think it's an important thing to do, to ask, is there an independent way to turn them on, right? So another way is they do turn on with age normally, but it's much more sparse in the brain. So the other possibility would be to take our, humanized TDP43 background that also has our mCherry reporter of replication, just let the brains get old and then look for cells where it's replicated and look surrounding. So maybe that's worth doing as well. Yeah. There was another question somewhere back.
You mentioned that, I think you mentioned there were spot foci of TDP43 in the nucleus, in the unaffected cells. Some smaller foci in unaffected cells, probably on the disappear. Do you know what those are?
Wait, you mean by unaffected cells, you mean the cells that were not expressing TDP43, we now see TDP43 foci nearby, is that Gene?
I think you said that, well, I understood it earlier on, there were some foci that
They never quite leave. In our hands, the cells probably die before they fully leave the nucleus, but we increasingly see more and more in the cytoplasm over time.
Do you know what those foci are in the nucleus?
The one thing I can say is that TDP43, when we look, I didn't show these, but when we look with really high resolution, there's this array scan that's like close to super resolution. It looks like there's structure within the nucleus. There are regions of the nucleus that TDP43 is not in. And we see that in cell culture as well. It looks to me almost like maybe nucleolus is excluded, but I don't have any markers to know that. It's just like a chunk of nucleus that looks pretty big that has no TDP43. Yeah. So I don't know.
Okay, well, let's thank Josh for a great talk.